Okay, so what is the sodium-potassium ATPase? What kind of molecule is it? It's a protein, okay? And you're right, it is a protein that has enzymatic function. And what is that enzymatic function? Hydrolysis of ATP, okay? This ACE indicates it's an enzyme, ATP is the substrate, and ATP can be broken down into ADP and phosphate. And why is that hydrolysis important? Well, the reason that hydrolysis is important is that it liberates phosphate groups. And what do phosphate groups do when they interact with proteins? When they phosphorylate proteins. They change its shape, okay? And then when the protein changes shape, the phosphate group might release. What does that do? The shape changes back again. So we can have proteins altering between different shapes or different conformations, just a fancy word for shape, as a result of this phosphorylation and dephosphorylation. Okay? This particular protein has a number of binding sites on it for other molecules. What kind of binding sites does this protein have? It has sodium binding sites. How many? 3. Potassium binding sites? 2. What other kind of binding site does it have? A phosphate binding site as well. What's the net outcome of this pump going through one cycle of operation? 3 sodium ions are pumped out, and 2 potassiums are pumped in. And in what kind of cells is this important? Well, as it turns out, it's important in a lot of cells, but the example that we've been using is neurons, okay? And when these 3 sodiums are pumped out and the 2 potassiums are pumped in, that neuron is restored to its quote-unquote resting state, and it has a membrane potential. Why? Because it's got an unequal distribution of charges inside and out. Okay? Do you know the name of pumps that create this membrane potential by creating this unequal distribution of charges? Not all pumps do this. This particular type of a pump that could do this is called an electrogenic pump. Sodium, potassium, ATPase is an electrogenic pump that creates a membrane potential. So now that neuron can be stimulated, and when it's stimulated, an action potential is generated, and we start getting this wave moving across the membrane, because what's happening? Ions start flowing across the membrane from high to low concentration. And in the process, the neuron membrane becomes depolarized. Okay? Well, then if that neuron's going to fire again, what has to happen? The sodium-potassium pump's got to pump the potassium back out again, the sodium back in again, and restore that membrane potential so it can be stimulated again. Okay? The big thing that drives this particular pump are a series of conformational changes. And when these conformational changes happen, what occurs? Well, one conformational change turns on the enzyme. It's initially inactive. And now it's activated to be able to do this. That's one result of a conformational change. Another change is that the pump can change orientation. And sometimes it can face outward, and other times it can swivel and face inward. Okay? Another change is that it can bind sodium or release sodium depending upon the shape of those sodium binding sites. Or it can bind potassium and release potassium. again, dependent upon the shape of those potassium binding sites. So all we need to do right now is just kind of follow the pump through a sequence and see what are the various initiating factors that cause conformational changes, and then what's the result of those particular conformational changes. And if you kind of go through that logically, then you'll have a good insight into how this particular sodium-potassium pump works. So that's what this diagram is all about here. Okay? So remember that the net goal of this particular cycle is to pump 3 sodiums out and 2 potassiums in to restore that membrane potential. So let's start over here. Okay? What does that sodium-potassium pump look like? Okay? Well, these are the sodium binding sites, these are potassium binding sites. The sodium binding sites are of the proper shape to bind sodium. They match. Okay? The pump is swiveled so that the open side faces inward toward the cytoplasm, because that's where the sodium has accumulated. It's rushed in during that action potential. So these 3 sodium binding sites can be occupied by sodiums because there's a lot of sodium there. But the shape of the potassium sites is not correct for binding potassium. So any potassium that's in here won't bind to those sites. It's not a good fit. Okay? This particular protein, its enzymatic capability is non-existent. It can't catalyze a reaction because it's the wrong shape. But when that 3rd sodium changes or binds, subtle changes in this activate the protein, and now it can hydrolyze ATP to form ADP and phosphate. And the phosphate group attaches to this molecule, this pump, and phosphorylates it. Okay? Phosphorylation of that particular protein causes some dramatic conformational changes. And one of the big ones is the pump swivels to face outward. Okay, so see the big conformational change that occurs here and here? Another thing that happens is the shape of the sodium binding sites changes. Sodium no longer fits into those binding sites. And it's going to be released out here. Why? Because there is less concentration of sodium out here. Okay? Notice that there's been a change in the shape of the potassium binding sites as well. And any potassium that's out here in the environment as a result of stimulation of the neuron can now bind to these sites. What do you suppose happens? That causes a change in shape of the protein. And now the phosphate group can no longer attach. A conformational change results in this phosphate being kicked off. Another dramatic change in the shape of the protein. It swivels back inward again. The shape of the sodium sites changes from this to this. Now these are going to be ready to bind sodium again. The shape of the potassium sites also changes, so potassium no longer can bind, so the potassium is going to be released back into the cytoplasm. We're back to starting point again. Okay? So do you see how the sequential binding and releasing of molecules from this protein stimulates a shape change that then makes the next step possible? And that's how this pump works to accomplish its goal of moving sodiums out, potassiums in, to restore that particular neuron to its resting potential. It's all driven by conformational changes. It's dependent upon ATP hydrolysis, but the purpose of the ATP hydrolysis really is to produce the phosphate groups. And they're really the initiating thing that causes the cascade of conformational changes that causes this particular pump to work. This is the same diagram, just shown in linear fashion as opposed to a circular fashion. Okay? So when you're studying for this, really what you should do is just use a diagram like this and say, what are the steps? Okay? And what does each one of those events then initiate as a next step? This is important background information to understand to help you figure out how this particular cycle really works. So it's not as complicated as it appears if you look at it in this kind of a logical fashion. Let's give something first. You really do learn something every day as you do more reading and talk to people. And until quite recently, probably last time I taught this course last year, my understanding was that the sodium-potassium ATPase was really only a big deal in neurons. But after talking to Dr. Rhodes about this, who teaches our physiology class, he set me straight, and now I realize that this particular pump is critically important in most cell types. What this particular pump can do in many cell types is generate a sodium gradient by causing more sodium ions to accumulate on one side of a membrane than the other. When we talk about a gradient, we're talking about a difference in concentrations. Well, anytime you have a high concentration and a low concentration, particularly separated by a membrane, that represents potential energy that can be used to do work. We're going to talk about this in another context when we talk about cell respiration. It's almost like water held behind a dam. So if you think of these sodium ions as like water molecules, if a lot of water is held behind a dam, that has potential energy to do work. In the case of a dam, generate electricity. So there are a lot of cells that are able to accumulate sodium ions on one side of a membrane. And then in the membrane, there are these channel proteins. And as the sodium flows across the channel protein from high concentration to low concentration, it can pull other things along with it. It can pull other molecules along with it. So many of these cells use this general mechanism of sodium pumping to build a gradient, and then use the gradient to move a lot of things across the membrane in conjunction with that particular membrane. In his physiology class, Dr. Rhodes refers to this as powering the battery to do cell work. You use the sodium-potassium ATPase to set up the sodium gradient, and then you use that to accomplish transport work. So this strategy allows you to use one general kind of pump or mechanism to do many kinds of cell transport. And here's a pretty staggering estimate. It's estimated that 30% of the cell's ATP in a typical cell is used for this one purpose of generating these sodium gradients to power transport of other molecules. A third of the energy that you produce is used for this one particular purpose. So if you go on and take physiology, you will hear more about this later on. Now, there's another kind of transporter that can be used besides the sodium transporter to power cellular work. In both eukaryotic and prokaryotic cells, proton pumps are also used to set up gradients to accomplish cellular work. A proton pump, instead of pumping sodium ions out of the cell, pumps hydrogen ions or protons. And so it creates a pH gradient. Okay? Remember, pH is a reflection of the number of hydrogen ions. So this would be a lower pH than this. That's a source of potential energy. We're going to see later on that H plus gradients are going to be used in the mitochondria as a source of energy to make ATP. That particular process is called chemiosmosis. There are some organisms, particularly plants, that couple a proton gradient with the transport of other materials into and out of plant cells. This particular process is called co-transport. So in co-transport, a membrane protein couples the transport of one solute to another. The example we're going to show you in just a second is coupling the transport of protons with the transport of sucrose, a sugar produced by plants during photosynthesis. Here's how that works. If a plant wants to accumulate a bunch of sucrose inside of its tissues as a result of the photosynthesis process, sucrose transport will result in cells accumulating sucrose at higher levels than it is outside of the cell. They're pumping the stuff into the cell for storage. So that's going to be an active transport, an energy requiring process. Okay? So what can plants do? They can couple that to a proton pump. So the energy is actually used to transport protons from inside the cell to outside, setting up this hydrogen ion gradient. And then if there is a co-transporter in the membrane, these protons will flow back down again their concentration gradient from high concentration to low concentration. Okay, that'll be spontaneous. And sucrose will be dragged along with it against its concentration gradient. Okay? So effectively, the transport of sucrose is being powered by ATP, but it's using this hydrogen ion gradient as kind of a driver for that. That way, we don't have to have a separate transport mechanism for everything we want to transport. We can use this proton pump to charge the battery, to provide the energy, and then couple that to the co-transport of sucrose or other molecules. And so it's kind of a way to have some efficiency and use one active transporter to drive several different processes. Okay, a couple questions for you. We have two solutions. Solution A, 0.2 molar glucose, separated from solution B, 0.4 molar glucose, by a membrane. And that membrane is impermeable to glucose. So which solution is hypertonic? A or B? See many responses. See a 2. See another 2. Another 2. Yes, solution B is hypertonic compared to solution A because it has a higher concentration of solvent. Which solution will gain water? The more hypertonic solution will gain the water. So if you had that membrane separating those two solutions, water would flow from the hypotonic solution to the hypertonic solution. Okay, in the sodium-potassium pump, what activates the ATPase enzyme? I made it easy for you. These are actually kind of in order of what happens. Okay, so the first really initiating event is a sodium binding, and that's what activates the enzyme to then hydrolyze the ATP into ADP. Okay, up to now we've been talking about the movement of small molecules across a membrane. Now we're going to shift gears and talk about the movement of large molecules, or perhaps even whole cells, across the cell membrane. And the ability for white blood cells to engulf bacteria is a great example of one cell pulling a whole cell of another type across its membrane. What do we call that process? That's phagocytosis. Okay, we're going to see that in just a second. Okay? So there are two general types of transport that are going to be involved in these large molecules. Sometimes this is referred to as bulk transport. Okay, so these are bulk transport. The two general categories are exocytosis and endocytosis. Exo means out, exit. Endo means to bring in. So exocytosis would be a form of secretion, and endocytosis would be an engulfment. Now, what has to happen here is that it's not just a permeability issue, okay? In the transport of small molecules, one of the big issues is that phospholipid bilayer permeable to the substance or not? These things are all so big that permeability isn't even a factor. I mean, nothing can cross the membrane. So to bring things in by endocytosis, or release them by exocytosis, the membrane is going to have to fundamentally change shape. And one of the things that happens is, in the case of endocytosis, the membrane is actually going to pinch in and form a pocket. That particular process is called invagination. So that would be an endocytosis. What's responsible for this? Well, the cytoskeleton is actually going to play a role in this. Because those microtubules and microfilaments and things can be attached to the cell membrane. And as they change shape, they can pull the membrane in and create this little pocket. And whatever happens to be outside in that area can be engulfed. And eventually the membrane can pinch off and seal itself, so we now have a little vacuole that is inside the cytoplasm of the cell. In order for this to happen, the membrane has to be very dynamic. It can't be this static structure like the dialysis tubing that you're using in lab. Okay? The dialysis tubing is basically like a piece of cellophane. And if you pull on it, it's not going to separate into two pieces that self-seal into two compartments. But a cell membrane can do that. One of the reasons that a cell membrane can do that is that it's fluid. And that's the next topic we're going to talk about, is membrane structure and what fluidity means. When something is excreted from the cell, a vacuole will have to fuse with the membrane, and then those two membranes will join together, and eventually part of the membrane will dissolve, and then the substance that were inside can be released. So these bulk transport mechanisms require a very dynamic fluid type of membrane. We're going to look at a little bit more detail about three types of endocytosis. And they are called pinocytosis, phagocytosis, and receptor-mediated endocytosis. So these are all types of endocytotic processes. Here's pinocytosis. Pinocytosis is sometimes referred to as cell drinking. So here's our cell, here's our membrane, here's the environment, and the little symbols just represent different molecules. Okay? As the cell membrane invaginates and a pocket forms, whatever was in the liquid surrounding the cell becomes trapped inside of that forming vesicle. And it's totally random. Okay? Whatever molecules happen to be in that vicinity are what get brought in. Eventually, the membrane seals itself off again so everything doesn't leak out. This vesicle completely seals itself, and now the stuff is in the cytoplasm. Okay? But again, what's brought in is random, just pure chance as to what was out here and how many molecules of this and how many molecules of this were in that vicinity. Here's an electron micrograph of pinocytotic vesicles forming in the small lining of a small blood vessel. You can see all these little vesicles here that pinch in and bring materials into the cytoplasm of the cell. So this is a non-specific process. There's really no control over what gets in. Phagocytosis is sometimes referred to as cell eating. Very similar phenomena to pinocytosis, except that large molecules tend to become aggregated in this particular location on the membrane. And when the membrane pinches in, these food or even entire cells, in the case of a bacteria, can be brought inside the cell in the form of a vacuole. So this is a way for the cell to get some large macromolecules. What's going to eventually have to happen to this food vacuole if the cell is going to use this as a food source?
Digestive enzymes.
Yep, it's going to have to fuse with a lysosome, and the digestive enzymes will start breaking this down. Okay. So this is an electron micrograph of actually an amoeba, which can do phagocytosis. The amoeba you saw in lab last week, engulfing a whole bacterial cell. This would be a very similar phenomena to what happened with your white blood cells if they were patrolling around your body and found a bacterium, and then they would engulf it to destroy it. Here's our third example. This is a process called receptor-mediated endocytosis. So phagocytosis by itself is pretty nonspecific as well. It just grabs whatever's out there. In receptor-mediated endocytosis, there are particular receptors on the membrane in the area where these vesicles start forming, where it pinches in. And the little red Y's right here represent particular receptors. Receptors only fit certain molecules, and so the purple triangle molecule fits the receptor, but the green circle molecule doesn't. So we get an invagination again. And instead of things just kind of randomly being brought into this forming vesicle, there's a little bit of specificity associated with it. And that is any of the purple triangle molecules will be grabbed by the receptors. Now, by chance, can some of the green molecules get in? Yeah. But this is going to favor bringing in the purple triangles. So there's not complete specificity here, but it gives you some additional control over bringing this in somewhat preferentially. So when they bind to the receptors, a vesicle forms. Don't worry about this coated pit, coated vesicle. But this receptor-mediated endocytosis gives you a way to bring some things in fairly specifically. Okay, there's a practical example related to this receptor-mediated endocytosis that's worth discussing. The condition is called familial hypercholesterolemia. Have you ever heard of it before? You know what the familial refers to? Tends to occur in certain families. So that would suggest that it has what type of a basis? Something genetic, right? So this is a genetic condition. And what does it have to do with? It has to do with cholesterol levels, particularly in your blood. In this particular genetic condition, what are the cholesterol levels? What does the hyper refer to? Elevated. Really, really high. And in particular, the type of cholesterol levels that get elevated in this condition are LDL cholesterol levels. You know what LDL stands for? Good or bad cholesterol? bad cholesterol is called low-density lipoprotein. There's also a good form of cholesterol called HDL. Okay? If your blood cholesterol levels of LDL get too high, one of the ways your body can respond is that receptor-mediated endocytosis can occur. So here's an LDL particle. Here are some receptors that recognize the LDL molecules. The receptors bind to the LDL particle. Endocytosis takes place. Receptor-mediated endocytosis takes place. And this cholesterol gets pulled into your liver cells. And then this cholesterol can be processed. And effectively what this does is it lowers your serum cholesterol level. Okay? So proper maintenance of your serum cholesterol level requires having liver cells with these receptors for these bad cholesterol molecules. Okay, so let's use the letter L to represent the normal allele for a receptor, and lowercase l for the mutant allele for the receptor. Okay. So there are 3 genotypes possible if we just consider this allelic pair. Homozygous dominant, heterozygous, homozygous recessive. People who suffer from familial hypercholesterolemia are homozygous recessive for those receptors. Both receptors have alterations in them, and they don't bind LDL particles very well. These individuals have 2 normal receptors. These individuals have some good receptors and some bad receptors. So which one of those would have the lowest cholesterol levels? Homozygous dominant, that's right. So they would tend to have low levels. Which one would have the highest? The one with malfunctioning receptors. And these would have kind of intermediate cholesterol levels. They keep revising the acceptable levels of cholesterol, but for total cholesterol, they're talking about under 200 now. Does anybody know your cholesterol level? You guys probably don't worry about that a little bit. People who suffer from familial hypercholesterolemia, instead of having total cholesterol levels of 200, can be up in the almost 1,000 range. Where people who are in the intermediate that are heterozygous can be, in that 400 to 500 range. A total cholesterol level of up near 1,000 is really, really dangerous. Okay. What's associated with higher cholesterol levels? Formation of coronary artery plaque, and then plaque can cause blockages in coronary artery vessels. Sometimes those blockages break loose and lodge in lungs and brain and cause clots, and can cause strokes and pulmonary embolism and all that nasty stuff. So this is a genetic condition where if you suffer from this condition because of this particular genotype, your prognosis for a long life is not good at all. Okay? And it all gets down to this fundamental, what receptors do you have in your membranes, and are they able to grab these harmful molecules out of your blood and remove them from your blood? It's all about your genes. Okay, last topic. We've been talking a lot about membrane function. And now we need to step back and talk about membrane structure and how that structure is related to the various functions that we've been discussing. One of the things we're going to spend a little bit of time talking about are various models that have developed over time that attempt to explain the structure of cell membranes. A model is really a hypothesis. And like any hypothesis, it is constantly being evaluated for validity. We're constantly doing experiments to gather data and then see if our data is consistent with the hypothesis, or this particular case, the model. We'll talk about a few people who have done and developed membrane models and how our understanding of those models has evolved over time. And the evolution of these models has been the result of new data that has suggested that the old model doesn't apply. Remember that when we have a hypothesis and we do an experiment, and if the data doesn't support that hypothesis, we should be revising the hypothesis. So that's what's been happening in our understanding of membranes is we evaluate models, new data suggests revision of those models, and that is an ongoing process. Our current understanding of membrane model structure is that the membrane is a fluid mosaic of lipid molecules, proteins, and carbohydrates. This particular model, called the fluid mosaic model, dates back to 1975. So this model wasn't even fully in place when we, Dr. Morden and I, were sitting in your place in undergraduate programs. Singer and Nicholson were responsible for proposing this particular model. And it was considered to be such a big accomplishment that they did end up receiving the Nobel Prize for it. I'd like you to understand and remember some names. I won't give you a lot of names, but I think that the people who make major discoveries in science are worth remembering. I'm not particularly interested in you knowing the exact date of something like that, although as I give you some dates, it's kind of interesting to put them in kind of relative order and sometimes associated with significant historical events. Oftentimes we use wars to be historical markers. This particular model, there's a lot of experimental evidence that favors this particular model. Does that mean that this won't be revised at some point in the future and replaced? Not necessarily. That's the goal of science. We're always trying to disprove our hypothesis or our models and see if we can find new evidence that requires that they be revised and updated. So basically, stay tuned. Okay, the first published model of membrane structure was proposed in the late 1870s. So, a little bit after the Civil War, to give you context. And a guy by the name of Overton made an observation by doing some simple experiments where he took red blood cells. And red blood cells are great models for studying membrane function and structure because a red blood cell is basically a membrane surrounding a bunch of hemoglobin. And it doesn't even have a nucleus, okay? He studied red blood cells and was able to investigate the rate at which various types of molecules moved across the membrane of that red blood cell. And what he found was that lipid molecules moved across cell membranes much faster than other molecules. And he understood chemistry. And in chemistry, I think I mentioned this before, there's this concept that life dissolves life. Polar substances dissolve really well in polar solvents. Non-polar substances don't dissolve well in polars. So gasoline, for example, and oil is non-polar. Water's polar. They don't mix, they don't dissolve. So he thought, well, if like dissolves like and lipids are able to move across these membranes fairly easily, the membrane must be lipid. Okay? A little bit after the turn of the century, a guy by the name of Langmuir said, well, it's not just any lipid. cell membrane lipids are primary a class of lipids called phospholipids. This is our shorthand representation of a phospholipid. Remember that a phospholipid is different from a triglyceride. Okay, remember this is a triglyceride. You've got glycerol, and then you've got 3 fatty acids that are joined by those ester bonds to the 3 hydroxyl groups of glycerol. And that these triglycerides can be fats or oils depending upon whether these fatty acids are saturated or unsaturated. Remember that from your mastery quiz? Okay. Well, in a phospholipid, one of these fatty acid chains is substituted by a very polar group, usually containing a phosphate. So this represents the polar phosphate group, and then these represent the 2 fatty acids. So this is why the phospholipid is represented this way. Polar groups are hydrophilic or water-loving. Fatty acid chains are nonpolar. They're hydrophobic. So these are kind of almost schizophrenic molecules. They're part polar, part water-loving, part nonpolar, water-hating. We call molecules that are like this amphipathic. The phospholipids are amphipathic. Well, Langmuir said, I know that the membranes by chemical analysis contain a bunch of these phospholipids. How could these molecules be happy and form a cell membrane if the membrane is basically separating 2 water environments? Because the outside of the cell, the tissue fluid, has a lot of water in it. But so does the inside of the cell, the cytoplasm. It's, 70-80% water. So how can this molecule be happy in 2 water environments? And his belief is that they form these layers called a bilayer. Here's our membrane, here's our out, here's our in. The polar heads are happy being associated with the water in the cytoplasm or with the water in the environment, but the core of the membrane is this hydrophobic core where water is excluded. Water tends to be pushed out of the inside of the membranes. Okay? So that's a phospholipid bilayer. Whoops. Gorder and Grindle actually said they were phospholipids. Okay, the next big change came in 1935, where Dapson and Dinelli said, well, we've done some more analysis of membranes, and there's something in those membranes besides phospholipids. The membranes also contain a significant amount of protein. They envisioned that the membrane was a phospholipid bilayer, and on the surface of that were layers of protein forming kind of a sandwich-like structure. So if this is our peanut butter sandwich, the bread would be the layers of protein, and then the phospholipid bilayer would be the peanut butter, or if you want a jelly sandwich, the jelly. Okay? And this persisted from 1935 until the fluid mosaic model, 1975. So this is what Dr. Morton and I grew up with in terms of this was the wisdom at the time of what cell membranes looked like. So here is our Dapson and Dinelli model over here, the sandwich molecule with our phospholipid bilayer, and then sheets of protein that covered the phospholipid layer. There actually was some good experimental evidence that favored this kind of sandwich type model. In 1950, the electron microscope started to become widely used in biology. And a guy by the name of Robertson started taking electron micrograph pictures of cell membranes. And he saw, when he looked at those, that the cell membrane looked like it was a 3-layered structure. He called that, don't worry about this term, he called that a trilaminate structure. So this was what the cell membrane looked like in those electron micrographs. And he said, boy, this makes sense. Here's our protein layer, our bread, another protein layer, bread, and the filling of this phospholipid bilayer. Okay? So some pretty, interesting experimental evidence. Well, there were several things over the years that didn't start adding up as new evidence was accumulated that just wasn't consistent with this particular model of membrane structure. And all those inconsistencies eventually were addressed by the proposal of this new model, the fluid mosaic model of cell membrane structure. And here's what our fluid mosaic model looks like. Much different view than the classic Dapson and Dinelli model. The key elements are still there, the phospholipid bilayer. Proteins, however, were not just confined to the surfaces of the membrane. Some of the proteins were actually inserted in the membrane. Some of them spanned from one side of the membrane to another. Another thing about this particular model is that the proteins were not in fixed position within the membrane. They're kind of like icebergs floating in a sea, that those proteins could move throughout this membrane from one location to another. And even more importantly, individual phospholipid molecules were not fixed in position. And that this molecule right here, a second later, might end up all the way over here. And this molecule might end up all the way over here. And that gave this membrane a fluid or dynamic characteristic as opposed to the static concept that's more like that dialysis tubing where the position of all the molecules within the membrane is fixed. So that's probably a good place to stop right now. So we will finish wrapping up talking about the fluid mosaic model on Thursday. And we made enough progress that we can devote Friday to review. Okay?